Good morning, everyone. My name is Tyrone Sweet, and today I'll be talking to you about a computational pipeline I built to mine Staph aureus for prophage. Currently, antimicrobial resistance, or AMR, is a worldwide crisis. In 2050, it's suspected to be responsible for roughly 10 million deaths. That's beating cancer, that's beating road traffic accidents, and so much more. It's a growing issue that we really haven't found a solid solution for as of yet. One thing that we do know is that genes are transducible by viruses. Genes are transducible by viruses. We understand that they're able to take in genes from the environment, from the host, as well as introduce them. And thanks to this paper by Creighton University School of Medicine, they proved that the MEK-A gene that's responsible for making Staph aureus methicillin resistant is actually transducible, meaning that if you have a virus that attaches itself to Staph aureus and has the MEK-A gene, now you have a whole line of MRSA. And because of this is why I decided to focus on prophages and figure out what's their function and role in Staph aureus. For this project, I roughly had 3 goals. The first thing I wanted to do is just see what all phages are present in Staph aureus. And once I figured that out, I wanted to kind of see what similarities and what all that they have in common, and basically determine their function as a whole inside Staph aureus. The workflow is basically broken apart into 3 different steps. And I'll go over each step in detail in a little bit. So the first step basically revolves around a tool called PhiSpy, and it's a prophage finding tool that was made in a collaboration by San Diego State and a few other universities. What's special about this tool in comparison to others is, one, it was open source. I usually prefer to work with open source tools because you get to see their code, their methods, and it allows you to really dig in and kind of see what's going on, which is great for verifying that what you're doing actually works. 2, this tool has a 6% false positive rate. So all the viral regions that it detects, the majority of them are actually viruses, and you're not just getting a bacterial region that was mistaken for one. And last, what I really like about PhiSpy is through experimentation, they figured out how to produce an algorithm that doesn't necessarily depend on known patterns from other genome sequences. They found out kind of what were the signs through recombination, and they figured out what were, some of the traits and breadcrumbs, as I like to call it, left by the recombination process. And this allows them to take almost any bacterial genome at a, generic level and mine it for prophage. And they added another addition to it where if you're working with a specific strain of bacteria for, like in my case, it's Staph aureus, you can specify that training set, which actually makes it a lot stronger in finding those different phages. I'm working with roughly 10,000 Staph aureus genomes that I got from NCBI, and after running them through this tool, I discovered nearly 200,000 viruses. And out of the 10,000 genome sequences, 9,996 actually had phages that were detected. Only 4 came up with 0, and it's not necessarily that they didn't have any phage, it's just all the genome sequences weren't whole genome sequences. There were some contigs in there, So with this particular tool, and it's a common problem with detecting viral sequences and viral regions in short genome sequences. And so far, each Staph aureus genome sequence has roughly 20 phages in common. There are a few that have like a lower number. It's not shown here, but I found one that only had 4 phages, which is kind of different compared to the rest of the data set, but yeah. So the second step in the pipeline is working with BLAST. For these 200,000 phages that I found, there's a lot of duplicates. So I wanted to kind of filter them by doing a network analysis using the different genomes as nodes and the BLAST hits as the edges. So once the clicks are formed, I'll see which viruses, each Staph aureus genome had in common, as well as, potentially find viruses that weren't discovered. Because in this scenario, if you have a phage that only matches with itself, it's possible that it's a phage that hasn't been discovered before, or a phage that's only in, that specific genome sequence. And once I find the unique phages, I'm going to compare them to a few known databases, such as VirusSite, to see, one, what all viruses do I have present? And 2, I want to compare it to the CARD database to see if which genes are also present in the phage. And the last step we'll be doing is working with another tool called Eggnog, which is basically a functional annotator. After I have all my phages and they're grouped, I'm going to run through this tool to build a database, and it should basically tell you what is the gene function that the phage is doing. And this is important because we do know that, as I mentioned earlier, the MEK-A gene is basically responsible for the methicillin resistance in Staph aureus. So figuring out, one, how many phages are already carrying this gene in known genomes, and 2, are there any other genes that are similar or have some kind of function that would also, I guess, have the same effect as the MEK-A gene. And just to, I guess, summarize what I did, basically my 10,000 Staph aureus genomes for phage, identified nearly 200,000 phages, and I classified the unique phages by gene function, and finally the database is used for, finally created a database for analysis. So I'd just like to, thank my PIs and my funders and collaborators. And are there any questions?